Hi everyone, welcome to another class on evolution. Hi everyone, welcome to another class on evolution. In the last video, we had left our discussion between mutationist versus the selectionist. So that rift was still there, that what is the main source of evolution, whether it is mutation or whether it is natural selection. Finally, this gap was filled and a new theory was proposed, and which is called the synthetic theory of evolution or neo-Darwinism. So today we would be discussing the synthetic theory. Now, theoretical geneticists like Fisher, Haldane, Sewell Wright, they provided some theoretical framework for the integration of genetics into Darwin's theory. And if you remember, in Darwin's theory, the main flaw was that he couldn't explain the source of variations. So they tried to integrate genetics with it, but the way they tried was too mathematical and beyond the scope of understanding of most of the people. Finally, a major breakthrough came in 1937 with the publication of this book, Genetics and the Origin of a Species by Theodosius Dobzhansky, who was a Russian-born American geneticist. And in his book, he was able to integrate genetics and evolution together. So finally, along with Dobzhansky, Ernest Mayr, who is also known as Darwin of the 20th century, Huxley, Simpson, and Stebbins, these people contributed for the final picture of evolution, and that is the synthetic theory. And it is true, they agreed that natural selection acts on individuals, but the impact of that selection would be seen in the population. So in a way, we can say that selection acts on individuals, but the population is the one which evolves. So finally, the smallest unit of evolution was taken up as population. Now, since we have talked about integration of genetics along with evolution, so first of all, we have to be very clear about certain terms that we'll be using again and again. So we'll begin with the vocabulary of genetic change. Some of these terms you have already studied with genetics, but anyway, this would be a quick revision. So allele, which is the different forms of a gene. One single gene which is coding for a particular kind of trait can exist in different forms, and those are called alleles. Then the allele frequency, that would be the proportion of that particular allele among the individuals of a population. Gene frequency, now sometimes these are confused, but gene frequency would be for the frequency of that particular gene only among the individuals of the population. Mutation, of course, that is a permanent inheritable change in the gene, such as if there is a change in the nucleotide sequence, or maybe the entire gene has been altered, it has been duplicated or deleted, that all will be mutation. Macroevolution, that is origin of new species and extinction of the old ones, and this we have seen through various evidences of evolution. And when evolution is at a small scale within the species level, it is called microevolution. Finally, of course, what is population? It is group of individuals of same species occupying a given area at a particular time. And within this population, we can have number of demes. These are group of individuals in a population sharing the same gene pool. So now we can say that species is a large unit, and within species there are number of populations. So population is a subset of a species, and within each population there can be number of individuals sharing the same gene pool comprising the various genes. So with this particular terms in order, now we can move on with synthetic theory. So according to the synthetic theory, these various theorists, they agreed that evolution is not because of one single factor, but it is contribution of number of factors. And these factors are gene mutations, change in the chromosome structure or number, genetic recombination, natural selection, reproductive isolation, migration, and lastly, hybridization. So in this, we'll take each of this factor one by one, and the first and second factor, that is gene mutation and change in the chromosome structure and number, both of them are mutations. The second one is chromosome mutation, where there would be change in the structure of the chromosome or the total number of chromosomes in the individual may change, either the number is reduced or it is exceeded than the normal number. So these two together are mutations. So mutations are the ultimate source of all variations that we know about, because for a particular gene, the different forms, that is the different alleles, they arise because of mutation. So many genes mutate 1 to 10 times per 100,000 cell divisions. And in viruses, this mutation rate is comparatively higher. It would be 10 to the power minus 3 changes per nucleotide per generation. But if we talk about eukaryotes, it would be 10 to the power minus 8 to 10 to the power minus 9 changes per base pair per generation. That will be the rate of mutation, which we can see is very low. But even such low rate of mutation can also create quite a lot of genetic variation. Let us take, for example, that in case of humans, we know that the genome consists of 3 into 10 to the power 9 base pairs. And in this, as we say, that let us say 10 to the power minus 9 changes per generation is possible. So total mutations in one genome can be this multiplied by 10 to the power 9. So this would be, these two will cancel out and this would be 3. Now this is in one genome, but as we know that we are diploids, so a zygote will get one type of mutations from one parent, from the other parent. So as a result, in zygote, the gametes which are fusing together, they would be bringing in total mutations of 6. That would be 6 mutations. And since we know that human population is 7 billion, so in 7 billion population, if 6 mutations per individual are occurring, so this would be giving us a value of 42 billion. So even though the mutation rate is so slow, still we will have such large number of variations, right? So from here we'll move on to other factors responsible for evolution. continuing with the factors responsible for evolution. So as far as synthetic theory is concerned, one factor was mutation, which is the source of discontinuous variations. Now moving on to another factor, that is genetic recombination. Now genetic recombination is due to crossing over during meiosis, and as we know that crossing over occurs during prophase 1 in the Pachytene stage, during which the homologous pair of chromosomes, they exchange the DNA among themselves. Now in this case what happens is that there is reshuffling of the existing alleles. No new allele is formed. Mutation can totally change the type of allele that one is having because of the change in the nucleotides, but here only from the existing pair of alleles there would be reshuffling, which is somewhat similar to as we shuffle a pack of 52 cards. However, this is also a source of variation, but it is continuous variation which would be occurring in sexually reproducing organisms, because meiosis occurs only during sexual reproduction. And once these homologous pair of chromosomes have been arranged on the metaphasic plate, they would be independently assorted, and out of so many different gametes, which two gametes are going to fuse together, even that would be a source of variation. So among sexually reproducing organisms, the entire concept of continuous variation, we can link it to recombination, independent assortment and fertilization. Now moving on to next factor, that would be selection, or simply natural selection. The same thing can be called selection. Now it is the only form that produces adaptive evolutionary changes. The environment imposes the conditions which determine the results of selection and thus the direction of evolution. So here we can say that selection is the director, or you can say it is the master of the orchestra. Natural selection is the director of the show, and it is going to decide that who is going to act in that show. So mutation can induce number of changes, but out of those changes which will remain in the population, that would be decided by natural selection. So remember that natural selection does not itself create any variations, but out of the various array of variations those are available, it would be selecting which are having the highest adaptive value, that is highest fitness. So in Darwinian terms, as we see, survival and fertility mechanisms that affect the reproductive success or promote differential reproduction, that's what is known as selection. Now this selection can be artificial or natural. Now artificial selection is where the plant and the animal breeders, they select for desired characteristics among the domesticated plants and animals, while in natural selection it is the environment which is playing the same role, where nature determines which kind of individual is most fit and it will allow only that individual to reproduce. Now here we have an example. Can you recognize that in this background there is one organism? Here we have a moth. Now this moth was quite common in the European habitat, and it is called peppered moth, which is light gray in color with small black dots. And as we know that these moths are diurnal. Moths are diurnal, so during the day they would be resting on the tree here, and the tree bark is encrusted with lichens. All over the tree bark we can see the lichens growing, and with the color of the lichen and the bark, the moth is totally able to camouflage itself. So the birds which feed on these moths, when they are flying nearby, it would be difficult for them to recognize this moth, which is totally blending with its surrounding, and that way this moth is protected. However, there was a mutation leading to the formation of a black colored moth. Now as we can see, this black colored moth is very distinctly seen over the tree bark, and for the birds it would be easy to catch this moth and eat it. So now we see there is variation, and variation is brought about by mutation. So here it was a dominant mutation because it is being expressed in the phenotype, so that means it is dominant. So from here we can see that this moth, which is scientifically named Biston betularia, they were well camouflaged as they rested on the lichen covered trunks. However, few moths had a genetic mutation which caused them to be black, and they are said to be the melanic form. And this melanic form of moth is also named Biston carbonaria. But any condition always doesn't remain the same. So what happened, that these melanic moths, although they were not well camouflaged and they fell prey to the birds, and one who is dying will not be living long enough to pass on the genes to the next generation. But these conditions change around 1760s when Industrial Revolution began in Britain, and Britain was the first country in which Industrial Revolution began, and it later on progressed to other countries of Europe as well. Now during this time, coal was the main fuel which was used, and there was excessive number of factories had come up, lot of coal was being burnt, and the levels of pollution became very, very high. Now the total scenario changed. If you remember, lichens are also pollution indicators. So when pollution levels are high, the lichens die, and the tree bark totally got covered with the soot, the black colored substance that comes out when we burn coal, that settled over the trees. Now you see the new picture. So what happens? This black moth is now totally camouflaged with the tree bark, while the white colored one is clearly visible. So for the predator birds, the one which is easily visible will be their food. So now these light colored peppered moths, they were the ones, they were eaten, and their populations started to come down, while the population of these melanic moths, it started to rise because they were protected from their predators. So what do we see? That the consequences of Industrial Revolution led to the selection of melanic moths, right? And this showed that in a mixed population, that means there should be variations. Variations are the raw material for evolution. If there are no variations, no evolution will take place. So variations were there, that is two variants, that is light colored and dark moths were there, and out of that, whichever was better adapted to the niche situation could survive and increase its number, while the other one decreased. But at the same time, no variant is completely wiped out. The variation remains, although the frequency of that variation would change. That's what selection brings in. And this kind of change was an anthropogenic change, that is induced by humans. It is the human activity, that is the burning of the coal, which led to increase in the pollution, and as a result one particular variant got selected. And in the 1950s, England passed a clean air resolution by which they reduced the pollution levels. Again, the environment became much cleaner, and now the story was reversed once again. That is, again the light colored moths, their population started to rise. Now let us use this information and solve a question. So here the change of the lighter colored variety of peppered moth, that is Biston betularia, to its darker variety Biston carbonaria is due to. So here the options are temporal isolation, genetic isolation, regeneration and mutation. So in this case, of course, it was a dominant mutation which led to the origin of dark variety, and that would be our correct answer That is option 1. Now we'll take some more examples showing selection. Now, industrial melanism was an example which showed that natural selection is occurring in nature. Now, similar example we find from the lab experiment as well, and that experiment is replica plating experiment. Now, this experiment was conducted by Lederberg and who showed experimentally that mutations occur even before adaptations. So what they did was that they developed a culture of Escherichia coli, which is a common bacteria, and from a single cell, it was transferred to a master plate, which would be having agar and a medium in it. And from their culture, once we spread it on the plate, what happens that each of the cell which is there will give rise to colonies, and these colonies can be seen by naked eyes because in one colony there would be about 10 to the power 8 bacteria or more. So if we look at the master plate here, this is with the E. coli colonies, and here there is complete medium, which means that all the different nutrients which are required for the bacterial growth are present in this medium. So agar is mixed with this complete medium, and over it the colonies are allowed to grow after incubating the bacteria for some time. Now they developed a very ingenious technique to make copies of this master plate, and that's what is called the replica plate. So for that, what they did was that a wooden block covered with sterile velvet was used, and this wooden block, when it is pressed onto the master plate, what would happen? From each colony, few bacteria will be transferred onto the velvet, and this block is then pressed on a fresh agar plate in which again complete medium is taken. So now from each colony the bacteria are transferred to this fresh plate, and over a period of time they will also form colonies which will be an exact replica of the master plate. Now in one of the replica plate, what they did was that along with the complete medium they also added an antibiotic. We can mention penicillin or streptomycin, any one of them. So let us, for example, we are taking penicillin. So what happened now? The replica plate was not an exact copy. There would be some missing colonies, and the colonies which are growing, those are the colonies which are resistant to penicillin. Now resistance in bacteria again is because of a gene, and that gene would be coding for an enzyme which will cleave the antibiotic. So when did this mutation occur? So certainly while the bacteria were dividing, somewhere that mutation cropped up, but now the situation when it changed, that is penicillin medium was present, only those bacteria got selected who had that mutant gene in them. So this again will lead us to the consequences that mutations are pre-adaptive, and it is selection which is directing that mutation, it is fixing that mutation in the population. And by fixing we mean that the representation of those mutants will increase. So mutations are continuously occurring in nature, but which one will get selected? That would be seen by selection. So in other words, we can say that mutation supposes and selection disposes. And finally, we can conclude that evolution is a stochastic process. Now, stochastic is a mathematical term which literally means that it is random and unpredictable. So this term here means that the process of evolution is random and unpredictable. So for evolution to occur, there is no goal. Evolution is never goal oriented. There are continuously changes occurring among individuals, and the environmental conditions are also changing. Out of that, some of the variants may get selected while others will be non-adaptive, and hence their population will start to decline. Now moving on from selection to other factors, that was reproductive isolation. Now reproductive isolation is, we can say, any mechanism which is preventing interbreeding between two species, and that maintains the purity of the species. So as we know by the definition of a species, that a species comprises members who can freely interbreed in nature and produce fertile offspring, but at the same time members of one species cannot breed with members of another species. So this reproductive isolating mechanisms can be either pre-mating or post-mating, and natural selection tends to favor the organisms that exhibit pre-mating isolation over those who are having post-mating isolation. So let us take each one of them one by one. So first of all, starting with pre-mating isolating mechanisms. That means which are preventing the mating itself between two species. So in this, one is ecological, or it can be called habitat isolation. So two species that occupy different habitats within the same area, so they won't encounter each other, and as a result there is no mating between them. Another one would be temporal, which can also be called seasonal. So we know that different animals in the wild have different breeding seasons. Same goes with the plants also, different flowering seasons. So at one time when one species is reproductively mature, the other is not. So mating between them is not possible. So that would be seasonal isolation or temporal. Next one is behavioral, which is also called ethological. Study of animal behavior is called ethology. So behavioral or ethological isolation. So there are very intricate courtship rituals among organisms with various species which are unique for them. So the mate from that species only would be attracted to that species. Like among frogs, their breeding season is same, that is rainy season, and it has been observed that 3 different species of frog might be going in the same area, in the same pond, but the sound that they produce, the male frog from its vocal sac produces a sound to attract its mate. That sound to us it seems the same, but it is unique for that species. So the female only of that particular species would be attracted to that mate. So that would be behavioral isolation. And lastly, mechanical in pre-mating category. Mechanical isolation, here mating is attempted. That means the breeding season is same. There is no problem with the courtship behavior. So mating does occur, but it is not completed because there are some morphological differences between their copulatory organs. So mating is not possible. It has been observed in the various species of dragonflies. The male dragonfly has a different shape of penis which is unique for that species only, and as a result it can mate only with the female of its own species. So that would be mechanical isolation. Now even if this fails, so then there would be the next category, that is post mating, after mating, the mechanisms which are preventing the mixing of these species. Now post-mating isolating mechanisms, first we can take gametic mortality, as the word itself shows that gametes will not survive if they are transferred into the reproductive tract of another species, and it has been observed even among some of the species of Drosophila like Drosophila pseudo-obscura, Drosophila persimilis, that if mating does occur, then the sperms in the female's reproductive tract get destroyed. So there won't be any fertilization, and again mixing of the species is prevented. Another is zygotic mortality. Now here fertilization also occurs, but either the zygote won't be able to survive or the hybrid, because since it's a product of two species, that individual is going to be called hybrid. So this hybrid, either during the development or just at the time of birth, they won't be able to survive. So we also use the term hybrid inviability. Now let us say even the hybrid develops and it is born. Then the next level, that is hybrid sterility. And these hybrids are quite strong, sturdy, but they are sterile. So that means further mixing of the species has been stopped. Like example, mule, which is a product of female horse and male donkey, and the reciprocal cross will be producing a hinny. And both mule and hinny are sterile. Then the last category, that is hybrid breakdown, or which is also called F2 breakdown. Now in this case, the F2 generation, the second generation, will be sterile or it will be too weak to survive. But the F1 hybrids are fertile. Now here, mule and hinny were F1 hybrids. They were sterile. So there itself at the F1 level, further mixing stopped. But here F1 hybrids are fertile, but when they reproduce, their next generation, that is the F2 generation, is sterile. And here we have the examples Tigon, which is a product of male tiger and female lion, and the reciprocal Liger. Now both Tigon and Liger are F1 hybrids and they are fertile. And they can interbreed among themselves, they can interbreed with any of the parent species, but always the F2 generation is sterile. So that prevents mixing of these species. Now the question arises that why did these two were able to produce fertile offspring even at the first go? So reason being that these Tigon and Liger are never formed in nature, because one is found in Asia, the other one is in Africa. They are geographically isolated, they never meet. It is only in the zoo under captivity that they have been bred, and this way we have come across the formation of these Tigon and Ligers. But in nature they are never formed. So that means still isolation works. Now let us take a few questions. Which of the following does not illustrate the phenomena of natural selection? So industrial melanism, certainly the peppered moths that we had discussed. Herbicide resistant weeds, that is certainly that when we use these chemicals, then the selection would be for resistant varieties. Antibiotic resistant microbes, that is a menace for the medical industry, because when we use antibiotics, some of the microbes do have resistant genes in them and they are the ones who are selected. And as they multiply, ultimately we have a larger population of such disease causing microbes to which we are not able to use antibiotics. And of course artificial breeding, that is not natural, that is artificial. So this would be our correct answer, and that is option 3. Now next one, using imprints from a plate with complete medium and carrying bacterial colonies, you can select streptomycin resistant mutants and prove that such mutations do not originate as adaptation. These imprints need to be used on plates with and without streptomycin. So of course we will be creating replica plates, one with the complete medium but without streptomycin or any antibiotic. The other one would be having complete medium and streptomycin. In the other choices, you see here there is minimal medium. Minimal medium would be a medium in which one of the nutrient or more than one nutrient is not given. So then it is not complete, rather we have removed one nutrient, so that is minimal. And only with streptomycin also will not show us the difference, and only without streptomycin will not show us the difference. So here the answer would be on plates with and without streptomycin. So this way we will come to know about the resistant and the non-resistant bacteria. And here our answer is going to be option 1. Now, let us move on to other factors responsible for evolution. Continuing with the synthetic theory, in which we had seen there were 7 factors in all which were considered to be responsible for evolution. So, in that we have already discussed mutations, gene recombination, natural selection, reproductive isolation, and now let us move on with the next factor, that is migration. Now, migration is again a very potent agent of change, and other than mutation, this is the only other factor responsible for changing allele in a particular population, because it can introduce a new allele in a population, and if individuals are moving out from a population, then a allele can be moved out as well. So, movement of individuals from one population to another changes the genetic compositions of the population. Now, here let us take an example. Here there are two populations, and in this population people are having straight hair. This is just an example to illustrate the concept of migration, and in this population the people are having curly hair. Now, we know that both of these traits, they are controlled by genes, and these two populations are separated. So, that means there is no intermixing in the populations. But maybe this barrier gets removed and people migrate from one population to the other. There would be mating between those members, and as a result, over a period of time, what we will see is that both the populations will start becoming homogeneous. Like here in this population, we can see even some curly haired people, while in this population we can find some straight haired people. So that's what is called gene flow, that is migration of individuals to and fro between populations, and that increases homogeneity in the populations. This is what is gene flow. Now, these migrations can be of two types, that is immigration, that is arrival of the settlers in a new region, and when these settlers come, they will be bringing in new alleles with them, and these alleles will be transferred to the existing population, leading to change in the allele frequency. And the other is emigration, that is the phenomena of leaving one's native region to settle to another place, and this also leads to change in the allele frequency. So, from one region, the individuals who emigrate would be immigrants for the other region. Now, the last factor, that is hybridization. Now, hybridization is considered as a minor factor, but it can also produce offspring of mixed ancestry. And hybridization is possible only when reproductive barriers are not fully established, they are incomplete. And when such species, they come in contact with one another, they may be able to interbreed with each other and produce fertile offspring, and as a result a new species may arise which will be called fusion species. So, by mixing of two species where the reproductive barrier was incomplete, a fusion species may arise, and if the reproductive barrier was strong enough, then hybridization won't be possible. So, we have seen that usually the species, they are separated by reproductive isolation, but if those isolating mechanisms are weak, only then hybridization would work. So, these are the various factors. Now, this entire synthetic theory, as we have seen, has integrated genetics along with natural selection, and this all was made possible only after the studies on population genetics. And population genetics is studied by Hardy-Weinberg principle. This is a very important tool to study it, and this was given by G.H. Hardy, an English mathematician, and W. Weinberg, a German physician, who developed this concept independent of each other, and it was published in 1908. Now, according to this Hardy-Weinberg principle, it is based on the concept that there would be no change in the allele frequency in a population over time. That means a population is going to remain in equilibrium. And this would be possible only if certain conditions are met. And what are those conditions which will maintain a population in equilibrium? Those are that there should be no mutations. There is only random mating. Random mating is that any individual of a population is free to mate with another individual of the population. There are no restrictions. Their population size is quite large. Then only Hardy-Weinberg principle operates. There is no migration. And last but not the least, there is no natural selection. So if these conditions are met, only then we will say that the population is in Hardy-Weinberg equilibrium. And we can state this principle that in the absence of the forces that we just now mentioned, which can change gene frequencies, a population may have any proportion of dominant and recessive traits which are going to remain constant generation after generation. And however, total absence of genetic forces is impossible, hence evolution will always occur. And whenever this Hardy-Weinberg principle is disrupted or the equilibrium is disrupted, that means evolution is taking place. And if these forces are not working, that means equilibrium remains established, and if it is established, that means there is no evolution. So in simple words, we can say that evolution is departure from Hardy-Weinberg equilibrium. Now let us take the mathematical calculation of this equilibrium. So for this, there is an equation for the gene pool, which is given as P plus Q is equal to 1. Now here this is a simple equation where we are considering one gene which is having two alleles, out of which one is dominant, the other is recessive. The dominant one can be represented with a P and the recessive one can be represented with a Q. So the sum total of all alleles for one gene in a population is going to be 1. If there are more than 2 alleles, then accordingly we can have P plus Q plus R. If there are 3 of them is equal to 1, the equation keeps on becoming more and more complex. But we'll keep the thing simple. Now, as we know, in diploid organisms, it is not single allele because each chromosome is in pair. So even the alleles are also in pair. So to get that equation for genotypes, we have to square this first equation. And when we square this first equation, we'll get P square plus 2PQ plus Q square is equal to 1. Where this P square would be representing, let us say, capital A, capital A. If I write, that would be the homozygous dominant individuals. 2PQ would be representing heterozygotes and Q square would be representing recessives. Because for one particular gene, if it is having 2 alleles, that is capital A and small a, there can be 3 different types of genotypes. And these 3 genotypes and their frequency, if we add, that will also be equal to 1. Now let us take one example here. Suppose a population has individuals who can roll their tongue. Now tongue rolling is a dominant trait. So there would be tongue rollers and non-rollers. So this is how the tongue on rolling appears. The people who can roll their tongue, the lateral side, they can be folded inward, and that's what is tongue rolling. So suppose in a population, the non-rollers, that means who cannot roll their tongue, non-rollers are 16 out of 100. So in this case, we have just mentioned that rolling of tongue is a dominant trait and non-rollers must be Q square, that is having the recessive trait, and if we convert it to decimal, it would be this. and that means Q would be 0.4. Now with this we can get from the first equation P, because we have seen that P plus Q is 1. So that means P is 1 minus 0.4, that would be 0.6. So now from this we can calculate homozygous rollers and heterozygous rollers. Homozygous rollers, for this we require P square, and P square we have just now calculated the value of P, and that is 0.6 whole squared. This will give us 0.36. And heterozygous rollers, this would be 2PQ, and putting the values, that is 0.6, Q is 0.4 into 2,